As a little codon chart to show you it will fold into the size of a credit card if you want to laminate it. Keep it in your wallet or purse, back of your phone, along with your periodic table of the elements, so you have it with you at all times, barring that you haven't memorized it. We'll get into that a bit later. Questions? Okay, 2 points of business. Point 1 is, when will our final be, and when will our lecture final be, and when will our lab final be? We'll have in each. So my goal is to be done when we leave here at Thanksgiving. And Tuesday before Thanksgiving would therefore be the logical choice to have our test and lecture. So that's my plan. Labs usually end the week before regular finals. So that would mean that the labs would be, the last lab would be the 16th and 17th of November, which would be the day for the lab final. So write that into your daily planners. 16 and 17, depending on which day you're there, for your lab final, and the 24th is our lecture final. Then you go eat turkey and forget about school until next February or whenever we come back. But don't make plans for spring break next year. Bastards. I mean, all right. So we want to finish talking about translation today. And we covered the initiation. And just to review that, for initiation, we have three initiation factors that do the same job in prokaryotes as well as eukaryotes. And that's 1, 2, and 3. 2 pretty much directly interacts with the initiator tRNA I, and 1 and 3 interact with the ribosome. Then in eukaryotes, you have 3 additional ones. EIF4, which is special because it interacts with the cap and the poly A tail, okay, and the ribosome. And then EIF5 and EIF6 are for scanning. So that the ribosome, which started at the very 5' end, scans along until it finds the Kozak consensus to start. So you have these 3 additional extra ones here in eukaryotes for those jobs. Those are things that don't happen in prokaryotes. You don't need a factor for. There is no cap or poly A tail, and the ribosome doesn't scan. It just, or presumably doesn't scan. It just finds the actual Schindel-Garno box, or if it does scan, we don't know how it does. So that brings us to, once you get to a start codon and everything gets assembled, what does it look like? So at that point, you're going to need to continue translation, and that is the elongation process, okay? So the very first amino acid, this is not showing the very beginning. So the methionine, and this is a bacteria because that's an F-methionine. That methionine would be on the tRNA right here, and the A site would be empty, and a new tRNA would come into that A site. It needs some assistance. So there is a factor for that called elongation factor 1. I don't know why they left the 1 off. It is a little different in eukaryotes and prokaryotes, but they both go by the same name. Okay, so we just call them EF1, elongation factor 1. Some nuances in prokaryotes, it's EF. It was originally called TU. In case the book says that, you know that's what it means. And in eukaryotes, it's sometimes called EF1 alpha. But they do the same thing. So there's really just one factor here to remember. EF1 brings in every new amino acylated tRNA and puts them in helps them fit into the A site. Okay? So once that's snuggled in, now the ribosome needs to join the peptide chain on the tRNA and the P site to the amino acid that's on the tRNA and the A site. So you're going to move this peptide, you're going to break this bond and reform it at the end of the next amino acid. Maybe. So that bond is going to break between the peptide and the tRNA and the P site, and the peptide chain is going to get hooked on to the amino acid, the end terminus of the amino acid on the tRNA and the A site. The little explosion should be up here, not down here. Okay. That is the activity of the ribosome, and we call that catalytic activity peptidyl transferase, and it's actually the RNA of the ribosome itself is driving that catalytic event. So we can call the ribosome, it's acting like an enzyme, so we use the term ribozyme for RNA molecules that work like an enzyme. So that is peptidyl transferase. That's the second part in the elongation event. Okay. Once the peptide chain is on the tRNA, now is located in the A site. So everything needs to scoot over by 1. Okay. When they draw this, they often draw these things to make it look like there's a hole there, right? And then we say it slides over. Well, how do you slide out of a hole? You don't. So it's not a hole. That's the problem. It's just laying there. So the whole thing just scoots over. That is called translocation. Oh, translocation. So everything will get scooted over. And the tRNA that was in the P site will be in the E site now. The A site gets moved to the P site, and the A site is now empty, waiting for the next new amino acid tRNA to enter there. Okay. This slide just showed you a more organic chemical close-up of what's happening. The nitrogen on the amino acid in the A site reacts with the carboxylic group on the amino acid and the peptide chain that's in the P site, and that's where you get the new peptide bond. So this tRNA is now, nothing's on it, and this tRNA has the whole growing peptide chain, okay? This shows you the very first amino acid. So this is methionine, and this is threonine, and that amino group interacts with that carboxylic acid group. That is catalyzed by the ribosome, okay? That makes that happen. So this new bond forms, and that bond right there breaks, and you get a peptide bond or amide bond. Chemically, it's called an amide bond, but because it's between amino acids, we call it a peptide bond. Okay. Now this one needs to be moved over here. Well, the tRNAs don't move. They're base paired with the mRNA. So what happens is the ribosome moves. The ribosome slides over one codon, and everything goes with it. So this goes to the E. The tRNA with all the amino acids on it goes to the P, and the A site is left empty for the next amino acyl tRNA, and it starts over. So you have 3 sequential steps in this. You have, I'll write them over here. You have amino acyl tRNA entry, that takes EF1. You have peptidyl transferase. That doesn't take anything, that's the ribosome. And then 3, you have translocation. And that takes another factor, EF2. EF2 kind of looks like an amino acid tRNA combo, and it sort of tries to budge its way in here, and that pushes everything over, okay? So this is the elongation factor 2. In prokaryotes, it's also known as EFG. Don't worry about that. We just call it 1 and 2 now, okay? So 1 for the first step, 2 for the last step. Peptidyl transferase is in the middle, okay? Don't confuse the word translocation with translation. Translocation is just the last part in the elongation cycle. This is going to continue for as many amino acids as the mRNA encodes, right? So if it scoots over, you're going to get another one. Scoot over, it's going to get another one. Scoot over, it's going to get another one, okay? Each time this happens requires some energy, okay? So these EF1 and EF2 proteins, all of these elongation factors and all of these initiation factors are all use energy, okay? They use GTP. So they're known as G proteins. So the G protein is going to hydrolyze or cause the hydrolysis of a GTP. So we can say this is an energy requiring step. Okay, so for initiation, it takes 1 GTP. For elongation, it takes 2. It takes 1 for the binding and 1 for the translocation. Okay, and for the release, termination will take another 1. Right, so all in all, it'll take 2 GTPs for every amino acid for the whole protein to be made. So the last step is when one of the 3 stop codons, also known as nonsense codons, enters into the A site. There isn't a tRNA that recognizes them. There's no tRNA that has an anticodon naturally to recognize them, but they are recognized by protein, and that protein is called a release factor. There are different ones in bacteria versus eukaryotes. Bacteria have two different ones. Each one kind of overlaps. So number one recognizes UAA, and number two recognizes UAA. So they both recognize that stop codon, and that is the most common stop codon in bacteria, as it turns out. But then they each recognize a different second one. So this recognizes the UAG one, this one recognizes the UGA one. So there's a little difference there. Now, I said this requires energy, and there's a third protein that actually does the energy requiring step, and that's called release factor 3. So RF stands for release factor, right? So in bacteria, you have 1 and 2 that actually interact, and 3 that is the G protein that stimulates the process. So 3 works with either one. And in eukaryotes, we have a little bit simpler situation. We have a single release factor. It'll recognize all the codons. And we have about equal, I think UGA and UAA are both a little bit more often found as stop codons, and UAG is less, but it's not by a lot. So it's kind of even out. This recognizes all 3 of them. And the second protein is just needed for the energy requiring step. So you have just a little E in front, again, to say it's eukaryotic. Okay, so how many translation factors are there all total? So you should make a list of them and know what each one does. Okay, it's like a listed here for prokaryotes and eukaryotes. For initiation, for elongation, and for termination. Okay. So there's for prokaryotes, there's 3, 2, 2. And for eukaryotes, there's 6, 2, 1, right? For those different steps. You don't need to know the 3. That one just has to be there. So that protein, the release factor, comes in to the empty A site when there's a stop codon there. It does actually bind to the stop codons. It forms hydrogen bonds, and it catalyzes, when it comes in and it hydrolyzes the GTP, it causes the bond between the tRNA and the peptide to be cut. Now the protein is free, so the protein is released, and that's why it's called a release factor. After that, the ribosome can try to shift over, but it's not going to be able to because it can't translocate when there's a protein stuck there. So the whole thing just falls apart, okay? So the release factor binds, the protein is released from the tRNA and from the ribosome, and then the ribosomes dissociate and are released from the mRNA, and the 2 subunits will come apart, and you get to go start all over again with the initiation factors can help the ribosome go find a new front end to the RNA. Okay. So multiple ribosomes can be translating on an RNA at the same time. There's no reason they can't. Each ribosome would have 9 bases inside of it, right? 3 codons. And we know that there's a little bit of extra space on the edge where another ribosome can't get. So there's about 21 bases total that get covered up on the RNA. So if your RNA is 5,000 bases long, right? Divide that by 21. That's how many ribosomes could theoretically fit on there. And this shows this is in a bacteria. So the DNA is this little thing you see going right through here, real faint. And RNA is being transcribed. And the RNA, as soon as it's being transcribed, has ribosomes that can latch onto it and start the translation process. So you can see this would be the beginning, and the RNA would be only long enough to fit maybe one ribosome, and now we can fit a couple. This piece of RNA this is a separate piece of RNA. So not only is there multiple ribosomes on each RNA, there's multiple RNAs being made from the same gene at the same time. So each one of these is an RNA. This one has a whole bunch. This one has even more. This one has a gazillion. Now, this one you can't tell as well, it's kind of wadded up, but here it is, almost to the end, and you can see there's a whole bunch of ribosomes on there. So these are called polysomes. When you have an RNA with a whole bunch of ribosomes on it, it's now called a polysome, and that's easy to purify. In fact, that's how RNAs were originally purified, is the ones that were being translated, you could centrifuge and the ribosomes would be big enough that it would pellet in the test tube. You can't see the protein being made, but you can see the ribosomes and you can see the DNA in this picture. All right, so a little review of this. You saw it probably in Life 1. You'll see it again if you're taking biochem, but we want to talk about it a little bit more, and that is there are 20 different amino acids, and what characterizes each one as being different is the chemical nature of its side chain. The side chains then allow us to classify the amino acids into some groups based on the chemical nature of that side chain. So this group here, for example, are the aliphatic amino acids. They have just a hydrocarbon side chain, uncharged, nonpolar, just carbon-hydrogen chain. Proline is an unusual one because the side chain actually forms a bond with the nitrogen on the amino terminus, which makes this a secondary amine instead of a primary amine, and that's going to have some unique chemistry. But then you have isoleucine and leucine and valine, alanine, and the simplest amino acid, which really just has a hydrogen there. It basically doesn't have a side chain, really, right? The side chain is hydrogen. What's hydrogen? It's just a proton. It's nothing. So glycine sometimes falls into that category, but glycine really doesn't really fit the mold for any other amino acid because it's just so simple. Another group would be the ones that have aromatic rings, and they're also a little bit non-polar, but they do have different abilities because they can form pi bond systems, right? So phenylalanine, tyrosine, and tryptophan. Tyrosine is a special amino acid because it has that hydroxyl group on it. It's the target of a lot of post-translational modifications. Okay, and we're not going to get into those, but a very important one is the addition of phosphorus. And that is a target for the addition of phosphorus to proteins. So all of these are nonpolar. They're all hydrophobic. They would rather not be surrounded by water. And a lot of times you're going to find them on the parts of proteins that interact with either another protein, so it's hiding it away from water, or with a membrane, a lipid. So it's holding the protein in maybe in the membrane. The next group here are those that are polar but are not ionically charged. Serine, threonine, cysteine, methionine, and cysteine is more polar than methionine. Methionine is weakly polar. And then, so methionine a lot of times gets thrown into that other category with the non-polar ones. You'll see some books put it there. Asparagine and glutamine have amides on the end, so they're polar. Then you have amino acids that have they're not just polar, they have an actual ionic charge. You have negatively charged base, ones that have acidic groups, like aspartic acid and glutamic acid. So they have acid right in the name. And then you have ones that have amines, which could act as a base and be protonated. So these are histidine, lysine, and arginine. And that's a typo right there. I wonder if I can fix that. Yep, the book bought the farm on that one. That'd be a big old fat eh. But you knew that because that looks like the wrong color entirely. Kidding me? Arginine, which I probably misspelled, didn't I? And then, what is its abbreviation? ARG and R. There we go. All fixie. So arginine, lysine, histidine, these are the basic ones, and aspartic acid, glutamic acid are the acidic ones. So it's all 20, I think. And so when these get put into proteins, they're going to be able to interact with each other, and the amino acid backbone, the amide linkage that forms, can interact, right, to produce the different layers of structure in a protein. So just the actual amino acid sequence would be called the primary structure, okay? And that's directed straight from the sequences of the codons in the RNA, which comes direct from the sequences of bases in the DNA. So that designates the primary structure. The primary backbones can form hydrogen bonds. Those can either make helices, which is an alpha helix structure, or a sheet, which is a beta structure. Sometimes it's called the pleated sheet. Sometimes it's called ribbon. Those structures, like here are some alpha helix and here's some beta sheets, they fold up within an individual protein, and then that is a secondary structure. This is just a local little area is a secondary structure. But if you consider all of them in the entire structure of the overall protein and how that forms into a three-dimensional structure, you have the tertiary structure, 3D structure of that enzyme. A lot of times, one functional protein is actually composed of multiple subunits that may be identical or may be different, depends on the nature of that exact protein, but that then becomes just a subunit of the overall functional protein, and we would call the combination of all the subunits the quaternary or fourth level structure, okay? And a lot of times this is written in shorthand. Primary, first degree, secondary, second degree, tertiary, and quaternary, right? Like those are first, second, third, and fourth degree levels of structure. Okay? When we talked about the RNA polymerase as a big enzyme that has several subunits. When we talked about DNA polymerase as a big enzyme that has several subunits, right? We're talking about the quaternary structure of that, all those subunits. Each individual subunit has its own 3D structure dictated by the localized secondary structure as well as interactions. To get the tertiary structure, you might need interactions, or almost always need interactions between the side chains, okay? And to get interactions between proteins, again, there you need interaction amongst the side chains. So again, we've now gone through the entire central dogma of gene expression, from transcription, RNA processing, and translation. And that would allow us to see the whole connection between genotype and phenotype, okay? So we have a particular gene here. This is the normal white gene, so it's wild type, and it's producing an RNA. That RNA can be translated. That protein, when it's translated, actually forms a pump that pumps molecules in so that the pigment can be made in the eye, okay? in the mutant sense, or the mutant case, either you make no RNA, or you make RNA, but the RNA has an altered sequence, and the protein has altered amino acids, and the protein doesn't work as properly. And in any case, you get either less pigment or no pigment, and that is a mutant phenotype, right? In the case of the no gene at all, no RNA, no protein, no pigment. This ties the genotype to the phenotype, okay? Now there's a lot of nuances with, is there just one way to mess up a protein? No, there's lots of ways to mess up a protein, right? And so each different way, each different actual mutation could have a different effect, and that would have a different phenotype. So you can have lots of different phenotypes from the same mutant gene. And that's why we have to do things like complementation analysis to determine, are we looking at a differently mutated gene, or are we looking at a different mutation in the same gene, which simply gives us a slightly different phenotype. Like with white eyes, with the complete loss of the function of this gene, you have no pigment at all. But what about eosin eyes? White eosin is the same gene, but it's not that the gene is lost, it's that the amino acid sequence is different. So the protein is different. So the protein functions differently. It doesn't pump in as much, and you don't get a dark red eye, you get kind of a pale orangey eye. I guess that's the end. So we need to talk a little bit more about how the amino acids are specified. This genetic code, and that's why I passed out to you a codon chart. If you didn't get one, come get one. So this ties in with our third part of gene expression. And we saw this diagram previously. You have your DNA, right? Double-stranded DNA. That's the source of our genetic information. One of those strands will serve as a template for the synthesis of an RNA. The RNA has the same nucleotide sequence as the coding strand. When the RNA is translated, those codons are read by the tRNAs, which are going to direct the linkage of the amino acids in the proper order to form properly functioning protein. So how do we learn the correspondence between a particular codon and the amino acid that its cognate tRNA brings? Okay, so the first idea was that this was a code, and there's a lot of history to this. This was born out at the time just after World War II, where a lot of the scientists who worked in this field of biology had been working in code breaking. They had been working on trying to, decipher the German and the Japanese codes and whoever else's codes they could break. And so the idea of, this is just the same problem we've been working on for 5 or 6 years. It's just now it's a biological code instead of an army code. So there's information in the DNA written essentially in the letters of nucleic acids, nucleotides, but it somehow has to correspond to an actual meaning, a real meaning of amino acid sequences. So what's the correlation? It's kind of a secret code. Well, if you have this correspondence, you have 20 letters or 20 amino acids that you need to specify, but you have only 4 symbols to specify them. So there can't be a one-to-one correspondence because that would only give you 4 corresponding amino acids. So there must be a combinatorial thing going on, either 2 nucleotides or 3 nucleotides or 4 nucleotides? Well, it's a simple matter of math, right? 1 to 1 would give you 4 possibilities. A 2 to 1, how many different ways are there to arrange AGCT in pairs of 2, where the order matters? Like AG is not the same as GA, right? So there's 16 possible combinations for that. How many different ways are there to arrange 3 nucleotides in order where the order matters? 64 different ways. Okay, how many ways do we need? Huh? 20. Is this equal to 20? Is this close enough to 20? Why can't we just round up? Nope. It's got to have it. Does this have 20? Like 3 times more than we need, right? And it's a log, you know, it's an exponential thing. It's like, nope, not even close. Ooh, really close. This would just work. This should work. And then like, now we got to go to the next step. And then the next step is like, boom, you know, it blows up. And you're like, why do we need all those extra 44 extra different ones? We don't need that. Well, it's okay. This idea that you would have 3 letters made sense from that mathematical perspective, but did it really correlate to what goes on? Because you could imagine a hybrid scenario where some amino acids might just need 2, but others might need 3. And that would give you some combination in between there that might be less than 64, but more than 16. So can we show that it really needs 3? And there is a set of experiments that was done with bacteriophage that allowed them to show that. And so we need to talk a little bit about how to alter sequences of DNA, which are going to alter the mRNA, which of course is going to alter the protein. So a set of chemicals called acridines and flavonoids can cause mutations where one base is added or one base is deleted. And if you work the system so that you use just the right concentration in the presence of, you know, the virus infection, you'll get like just one insertion in the whole genome of the virus instead of a whole bunch. And you don't want too many changes because too many changes is impossible to really figure out. So they sort of titrated it so they could know that just this tiny little bit of flavonoid, like this is something like riboflavin or those kinds of chemicals, or acridine orange, some of these things that bind to DNA, they just throw a little bit in, tiny little bit, and then that'll cause 1 or 2 mutations. So what happens if you add a base? If you were reading this thing in groups of 2 or groups of 3, adding one base would change the neighbor, and you would get a different read. It would make a different message. Taking one away would do that. What they found was that if they had inserted a base in one mutant and deleted a base in another mutant, and then got those 2 mutants to undergo recombination, essentially the addition was canceled out by the subtraction. Anytime you added 1 and then subtracted 1, you're okay. Added 2, subtracted 2, you're okay. Added 3, subtracted 3, you were okay. Okay. So the only times it messed up is if you added 1, took away 1, added 2, took away 2. If you did that, you had a mutant. If you took away all 3, you were fine. If you added all 3, you're fine. So Francis Crick and some of his buddies were ones to do these experiments. So they're looking at a bacteriophage, and it lyses the cells, and it either lyses really, really good, or it doesn't lyse so good. And so they can tell the difference. And so R is really good, and R2 is not so good. And in the addition of one base, you have a problem. They didn't know what bases they were adding, by the way. And so they were doing these experiments in the late 40s, early 50s. And no one knew what the heck they were doing until the late 1980s. 30 years later, when we actually got the technology to go and sequence these genes, some curious students said, I wonder what they were really doing. And they tracked down these old mutants that were still stored away in a freezer in England, and they sequenced the DNA in them, and they found that this is where the actual changes was. So this one actually had an extra A stuck in right here. And so the amino acid sequence is supposed to be what's listed here, but now you can see it's quite a bit different. And as a consequence, that protein doesn't work. If you put one in, so they took this mutation and they took this mutation and they got them to undergo recombination. They basically infected the bacteria with both viruses at the same time. And some of the baby viruses that came out of that, not really babies, but you know what I mean? Some of the progeny actually had a combination where they added an A, but they took out the G, and now they're right back to where they started. Just one amino acid is different, and that amino acid is not important. So these work just fine. So adding one and subtracting one works just fine. They did that in two different ways. Taking away two, that's a problem. It's not on here, but they had one where they added two. That was a problem. But if you take away three, like all three of these nucleotides right here, for valine, just gone. Well, that valine's not so important. So the protein still works. Anytime you had a combination that you took one away, added one, so you basically restored that system, or took away all three, it worked fine. So that gave a lot of support to the idea that 3 nucleotides were a codon. And that's true. We know that now. We have the codon chart and everything. Okay, how is that code read? How does the ribosome read it? How do we know that the ribosome doesn't just jump around? Well, if it just jumped around, those mutations wouldn't have the effect that they have. Okay, so we need to talk a little bit about what we call a reading frame. So if we took a sequence of nucleic acid, this is not nucleic acid, but this is a sentence, and it's buried within some other letters. And we're going to read it. It's made up of all 3-letter words. So where do we start? Do we start at the very first letter? Okay, so start at the very first letter and read that. You can make up the sounds you want. Bro, one, no, this, eka, this, it doesn't sound like English to me. Okay, somebody in probably going to watch this on YouTube and go, oh, they can speak Urdu or whatever it is, you know? It's like, okay, great. Not my intention. So what if we started not with the first letter, but the second letter? Ro. Man, that sounds a lot like English to me. So that might make sense. I mean, ro and whatever, okay? So we're listening to a Joe Biden speech, but it's just maybe part of it will make sense, maybe part of it won't make sense. So there is a message. We just need to start at the right place to read that message. Okay, the place you start, the nucleotide you would start at, are you starting at position 1? We would call that frame A. If you start at position 2, we call that frame B, right? And you're going to just keep in those groups of 3, or obviously the third one we'd call frame C. Okay, where's frame D? Well, you're back to frame A again, right? So if you scoot over one more, you're now at the 4th nucleotide, which is back to the same grouping that you have with frame A. So there's only 3 frames, A, B, C, when you read one strand of the nucleic acid like this, right? So if we look at frame B and we said, well, row and, that doesn't make much sense. But if there was one particular word that meant start the message here, and another particular word that meant end the message here, then we could go and just read that part. So if the beginning word was always now, right? Okay, now the cat saw the banana and did not run. And then the last word is in, but you don't read that one, okay? So think back to some Western that you've ever seen on TV or at the movies, and they're sending a telegraph message, right? They're sending the telegraph, and then they're reading it back, and it's like, you know, Betsy, let the cows out. Stop. Right? The stop is not read. What is the stop? It's a period at the end of the sentence to let you know that's the end of the sentence, right? So we don't really say the stop codon. The stop codon doesn't encode an amino acid. The stop codon just causes the process to terminate, right? But there has to be a stop codon there. So we do include the stop codon in the reading frame, or at least I do. There are other people who won't, but you're in my class. So the stop codon is the last codon of the reading frame. It isn't read by a tRNA, but it's read by the termination factor, right? The release factor. So we have the idea of a reading frame. Now, could this reading frame be in a different frame? Sure. If we didn't have a G, right? If there was no G, where would our reading frame be? There's no G. Now, where's our reading frame? Oh, it's frame A. All right, there's nothing magical about A, B, or C as the frame. It's just that it's a construct that allows us to understand which nucleotide we should be starting with, or which nucleotide the ribosome is going to be starting with. And then here's the kicker. Once the ribosome starts, it will not alter its 3 amino acid grouping. If an insertion of a nucleotide happens, SOL, the ribosome is not going to know which nucleotide it should skip over, right? There's no way it can know that. It just reads 3s. So if you added 1, it's just going to read the wrong 3. If you took away 1, it's just going to read the wrong 3, right? So once a reading frame is established by the ribosome, it's going to stick in that reading frame regardless of any alterations to the sequence. Any insertions in here are not going to cause the ribosome to move. It's just going to cause the ribosome to read exactly the way it was reading, but not read the right thing, okay? So again, here, the third one just shows you if you took off the first 2 letters, now you would start in the third reading frame. So this brings us to categories of mutations where nucleotides have been added or subtracted. Those are called frame shift mutations, okay? Because the correct reading frame, the one that the original sequence meant to encode, is now found shifted. If you were supposed to be in A and you insert 1, it will move to B. The actual correct amino acids would now be found in B. But what does the ribosome do? If it started in A, it keeps going in A, even if the correct sequence should be what's found in B, all right? So if we now look at this, I'm going to look at it with actual DNA sequence or RNA sequence. It doesn't matter. I have DNA sequence here because that's what I loaded from the computer. So don't worry, T's are U's and U's are T's. We're all one big happy nucleotide family. All right, so if we looked at what does ATG code, what does CAG code, what does GCG code, right? And that's where you can use your handy dandy little codon chart, okay? So let's take just a second to look at this thing, and we'll talk a little bit about how you use it to determine that from it. So the first nucleotide of the codon, okay, and that would be in this grouping, like A. So you find that on the left, okay, it's third group down. Now you go across and you go, well, what's the second one? It's a T. So go up here to the top. Oh, there's not a T. U, okay? So you look in the first column, and then the last nucleotide is a G. So you go over to the far right side and you look for the G. So you should be in the A group in the first row, and then in the third thing, and that's an M, methionine, okay? And this is just, I put the single letters in here to make it small. Over here, if you don't know the single letters, is the secret code to go from single letter to 3-letter to the full word. Okay, so you can find all of them. So this would be methionine, glutamine, alanine, tyrosine, so on and so forth. Okay, so the original reading frame is frame A because we find the start codon here. What if we insert a nucleotide? No, actually we deleted a nucleotide, so we took that T out right there. Is that going to change the sequence of amino acids? Is it going to change which frame is the reading frame? Well, now we find that ATG lines up with frame C. So it did, in fact, change the reading frame. Are these amino acids the same as those? They are. So it did not change the sequence of the reading frame, it just changed the frame. Okay, so this is a mutation that happened where? Outside the reading frame. It happened in the 5' untranslated region. I guess purple is not the color if you want to erase. So if you have a change in the 5' UTR or the 3' UTR, there is going to be no effect on the amino acid sequence because that's not where the amino acid sequence is. So the only place you can have an effect is in the open reading frame. And if you have a change there, you're going to affect the amino acid sequence somehow. So let's look at those conditions here. Once the ribosome actually starts and it starts translating here, what if you change a nucleotide somewhere in the open reading frame? So what did we do? We have 2 A's up here, TAA, TAA. So we added an A. We added a base, right? So where does our reading frame go? It goes right here. It's in A. So the ribosome is going to go MQAYL just like it was before, but now it's going to read AAT, which is a different amino acid entirely. And then everything else after that is a different amino acid. In fact, it just keeps going because it doesn't even read the stop code on it. It makes the wrong amino acids and it makes a bunch more amino acids than it ordinarily would. This is called a frame shift mutation, okay? A frame shift mutation. Where are the correct amino acids located? They're actually down here, okay? So if we skipped over to ATC, we would get the right amino acids again. So the actual amino acids shifted to a different frame. That's why this is called a frame shift. The ribosome does not shift, and therefore the ribosome makes an improper protein, a totally mutant protein. Frame shift mutations are extremely deleterious on the protein. They almost always totally jack up the function of the protein. The only time they wouldn't is if they happened, right almost at the very end of the protein, so there wouldn't be very many amino acids different, okay? And most of the protein would be there, and probably it could do its job. But even then, they could still jack it up. So frame shift mutations are very problematic. You don't want that to happen. Again, it can only happen if the insertion or the deletion happens in the open reading frame okay? If it happens outside of the open reading frame, it might cause a different kind of mutation, but it won't cause a change in amino acids. Okay? It might cause the RNA to get destroyed, or it might cause the RNA not to be spliced properly or whatever, but it won't cause the protein to change. So here's another one. What if we change an amino acid? Oh, look, I made it a U down there. What if we change an amino acid, but we don't change the reading frame? So what if we just change, this is called a substitution. So we had a sequence that was AAG before, that's what it started, and now it is UAG. So the A got changed to a U, or I could have put a T there. UAG is one of the 3 stop codons, right? Look at your codon chart, and you'll see over there that it says stop. U, and then A, and then G, stop. Okay, and tiny, tiny little letters to fit in there. A lot of times we use an asterisk to represent a stop codon. That's just, you'll see that. So this protein is totally jacked, right? You only get 6 amino acids out of however many there were supposed to be, and that's not going to work. So this is called a nonsense mutation. When you take what was originally a codon that specified an amino acid and alter it so that it now is a stop codon, that's called a nonsense mutation. What if you change a nucleotide? Okay, so we had AAG again, same one, but now it's changed to a GAG. Okay, so the reading frame is not changed. The ribosome reads it, but GAG codes for glutamic acid instead of lysine. That's K. So you get a different amino acid than you had before. Still get the protein. Protein's the same length. One amino acid is different. That might be a big deal. It might not be a big deal. Kind of just depends. You have to look at that as a case-by-case basis. But we call that a missense. Everything still works. The message is just off by one word, okay? And out of context, you could still figure out what the message meant, right? But it depends. Like if you're reading a true-false question and you misread the word not, that's going to give you the opposite answer, isn't it? So sometimes those little words are important. And so this example right here is probably a big deal because this amino acid, do you remember? Lysine is one of the, is it charged? Is it nonpolar? Or is it polar non-charged? It's polar and it is charged. And what charge does it have? Positive. And this is glutamic acid. So is it charged? And it has a negative charge. So you went from a positive charge to a negative charge. That almost always is totally bad news for the protein because that positive charge probably was doing something that you need a positive charge for. And if you flip that to a negative charge, it's probably not going to be able to do any of that stuff anymore. So that could be a very bad change. Okay. If you changed it from lysine to arginine, that's one positive charge to another charge. Most of the time, that won't cause a problem because it's called a conservative change. So a lot of mutations are, if you look at it, you go, well, I just changed one amino acid. Well, what was the chemical nature of that amino acid? If you change the chemical nature of the amino acid, it's almost always a very serious mutation to the protein. If you conserve the chemical function, if you go a polar for a polar, it's probably okay. If you go charge for the same charge, probably okay. If you go nonpolar for another nonpolar, probably okay. Okay, it still make a little difference, but it's probably going to be kind of okay. So how did people figure out exactly what the correspondence was, right? You had 64 choices to match up with 20 amino acids, and there has to be some ability to have like punctuation, they called it, right? Like the stop codon and a start codon. Well, if you can have a lot more potential than need, that's going to allow for what we call degeneracy. Now, if you're a code breaker, degeneracy is like awesome, right? If there were like 8 different words that you could use for, you know, bomb, then it would be a lot harder for the people breaking the code to know when you were saying bomb, right? Like in a classic example of how they broke the German code was like there was some place where the first word every day was good morning. And they're like, okay, so good morning was 531 today. Good morning was 621 yesterday. So they could find that first message and they knew what good morning was, and they could figure out all the rest of the message from that. Okay, so if they'd have used 6 different words for good morning, then I wouldn't have had that problem and the Germans might have won the war. But thank God they were friendly, good morning people. I mean, they were still Nazis, but whatever. So the degeneracy here is you got 6 different codons for some amino acids. You got 4 different codons for a bunch of them. And most of them have at least 2, and there's just the occasional methionine and tryptophan for us that only have 1. But for other organisms, they have 2 methionines and 2 tryptophan codons too. So the general rule is 2 codons at least for every amino acid, with some having up to 6. So how did they figure that out? Well, they did 3 different little techniques. The first technique was they figured out that they could synthesize a stretch of RNA, and that stretch of RNA they could put in a test tube and with some ribosomes and with tRNAs, and the tRNAs would have radioactive amino acids on them, and they could figure out which radioactive amino acid had been incorporated into a protein. So they could figure out what sequence of RNA corresponded to what amino acids. And the first ones they did, they just took an RNA that had only 1 nucleotide, like it was just all A. So it didn't matter where you started, right? You started the first 3, second 3, wherever, you're going to get the same amino acid a whole bunch of times. So they did that with all 4 of them. It took a while with G. G was a little hard, but they did it with all 4 of them. And the second strategy, they took just 2 nucleotides. They could build that in a test tube. They could take those 2 at a time, and then they would take those 2 and they would hook them together. So you could create like GA, 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 or UC, 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 right? Over and over. And then they would do it with 2, or they'd do it with 3, or they'd do it with 4, and they would put that in the same test tube and see what they could get with the amino acids. So that allowed them to figure out some. And when they put these 2 pieces of information together, then they could actually figure out a whole bunch. And finally, one group of people took 3 nucleotide long pieces of RNA and interacted them with tRNAs, and then got that to interact with the ribosome, and then trapped the ribosome to see what amino acid was trapped. That was the most complicated of them. And this is what it took to prove that no tRNA actually interacts with the stop codon, okay? Because if you put a stop codon in this, you just don't get an amino acid, but it doesn't stop either. It still gives you stuff. You don't know that it didn't stop. You just get fewer amino acids, right? So the first method is shown here. So if you take a homopolymeric RNA, all the same nucleotide, right, and let it get translated, here's what they found. All U's give you phenylalanine only, incorporate only phenylalanine, A's only lysine, C's only proline, G's only glycine. So there's 4 codons. UUU has to be phenylalanine. GGG has to be glycine. CCC, proline, AAA, lysine. So you have 4 of them. Yeehaw, 16 more to go, right? So the next experiments that they did was a little variation on a theme. They said, let's just don't put one nucleotide in. Let's put in two nucleotides, but let's don't put them in exactly equal. Let's do a whole bunch of ratios. Like we can do 50-50, 60-40, 70-30, 80-20, 90-10. Do these different ratios. And then statistically, there's a likelihood of a particular three-letter combination happening, right? So in this example, if you did a 70-30 mix of G and U, then what's the likelihood of getting G and a G and a G? Well, statistically, it's 0.7, 0.7, 0.7, or 34%. What's the likelihood of getting three U's? Only 3%. What's the likelihood of two G's and a U, or one U and two G's, right? So they can figure out a percentage likelihood, and then they translate this business. And as it turned out, they would get different percentages of amino acids incorporated, a whole bunch of different ones, and that would allow them to make some correlations. So they already knew UUU was phenylalanine and GGG was glycine, right? But when they did the translation, they got more than 34% glycine. So what does that tell them? It tells them, dude, we already know GGG is it. So there's 34% of it. So how much is left over? 15%. Oh, look, three of these could also produce 15%. So that means that glycine might be GGU and GGG, or UGG and GGG, or GUG. So anytime you had two G's or three G's, you might have a glycine. How about valine? How about, well, let's go down here, phenylalanine. Only 3% was phenylalanine. And so what that meant was the only one of all of these that can make phenylalanine is UUU. None of them other ones could be phenylalanine. 6% is leucine. Well, there's three possibilities. So leucine has three possibilities. Cysteine has three possibilities. Okay? So they weren't really getting exact correspondence, but they could narrow it down, right? Cysteine now has three choices instead of 64 choices to be, right? Glycine, we now have one known and three possibles, right? So could they compare that data with other data? And they did. So they compared that data with the second method, where they took dinucleotides and trinucleotides and hooked them together. So right, if you create a whole bunch of UC, you can make UCU or CUC, 50-50. And what they found was they get two amino acids encoded, serine and leucine. So one of these makes serine and the other one must make leucine, okay? Now, can you put that together? Well, if you put that information together with this information, let's look at this. So here they have a cysteine. The choices for cysteine are either UGU or GUG. The choices for cysteine here are UUG, GUU, UGU. Which of these choices is also here? Only one of the three choices, right? And only one of these two choices, and it's the UGU. Boom, UGU must be cysteine, and GUG must be valine. So, oh, look, we had a valine. So we know GUG is one of them. There's still possibilities, but we can cross off UGU. So now we've narrowed valine down to one known and two possibilities. So these scientists did this. It took them like 5 or 6 years to go through all these possibilities and work them out, but they did. The very last experiment was to take little 3 nucleotide triplets, let them interact with tRNAs in a test tube. The tRNAs had radioactive amino acid, one out of the 20 would be in there radioactive. Then they have to let that interact with the ribosome, because you cannot trap this by itself. It's too tiny. But if you put it in a ribosome, it becomes huge. And then you can trap it. You can capture it chemically. And then you just look to see what was there. So you have 20 test tubes, and you do this 20 different test tubes. You put the same thing in 20 tubes, but in each tube you have a different radioactive amino acid. And you just look for the one that actually becomes radioactive in the ribosome. And then you know, okay? So they do it with a few controls like UUU. You know it's supposed to be phenylalanine? Yeah, it is, okay? When they would try this with the stop codons like UAA, nothing ever got trapped. Nothing ever got trapped. So UAA does not correspond to any tRNA. They said, so what if it was a stop codon? Okay, that makes sense. Okay, so through all of those efforts, we get our universal codon chart. Okay, and this is similar to the one I passed out to you. They have rewritten each codon in its box. So you would start with the first nucleotide as a U, second nucleotide is a U, third nucleotide is a U, right? And then they just rewrote it, which would take up a huge space. I mean, you'd have to have a backpack to carry this codon chart around in. See, it's huge. That was a joke. So, and then they write the 3-letter code here. And you'll notice, for example, phenylalanine has 2 codons. Leucine has 6 codons. Threonine has 4. Proline has 4. Serine has 4 here and 2 more over here. 6. Arginine has 2 here and 4 more here. 6. So arginine, serine, leucine, they have 6. A bunch of these, valine, alanine, threonine, proline, and glycine have 4. And then, you know, 2, 2, 2, 2, 2, 2, 2, 2. In some organisms, methionine has 2 and isoleucine has 2. In us, isoleucine has 3 and we just have 1 for methionine, but that's something that varies. In some organisms, these are both tryptophan and these are stops. Okay, instead. So this is what we call the universal. It's not, but it's human. So yeah, right? If the yeast had discovered this, then we'd be the exceptions and they'd be universal, but they didn't do it, little suckers. They left it to us. Now you can present this in other ways. I like this one, this circle, right? But you can't fold a circle up very easily and laminate it, but it's kind of cool. So first, second, third from center out, and then it allows you to have both the abbreviations there. And you can see from the outside ring kind of how many codons there are going to be for that thing. So if it's a wide one, you got 4, right? If it's a skinnier one, you have 2. Real skinny, you got 1. So that's another way of presenting this in there. Now I've looked for apps that were free because I'm a cheapo, and I haven't found a free one. There may be ones now. I haven't looked in a long time because I have this. But the apps I used to find, you had to push in a codon, or you had to type in the amino acid and then it would give you the codon possibilities. It wasn't just a chart that you could look at. So you had to, like if you were going to work on a problem and you were going to like, okay, so you'd have to type in every single freaking codon and then it would tell you, rather than you just being able to look and then look and then look, okay? And after a while, you notice, well, if these all have the same amino acid, then is this really a 3-letter code? It's not. What do they have in common? It's really a 2-letter code, isn't it? AC. AC equals threonine. It doesn't matter what the third nucleotide, and the third nucleotide is the wobble base, right? It doesn't really matter. How about proline? What is the genetic code for proline? CC, right? CCN. The third nucleotide doesn't matter. How about serine? It has 2 2-letter codes, UC and AG, right? But here the third letter matters because AG could also be arginine. So sometimes the third letter matters, sometimes the third letter doesn't matter. So it's really kind of a hybrid between that 64 and 16, right? Possibilities. So for some of these, right? GU is valine, GC is alanine, AC is threonine, CC is that, and UC, and so on. So for most of them, a 2-letter code, well, it lets you look at the last nucleotide and go, oh, the last nucleotide is a pyrimidine, the last nucleotide is a purine. Pyrimidines, purines. Pyrimidines, purines. Pyrimidines, pyrimidines, right? So when it's split up into groups of 2, it's because the last nucleotide is either a pyrimidine or a purine, okay? So something that scientists have been doing lately, which is kind of cool, is saying these 20 amino acids are not the only amino acids that you could make. There are other possible amino acids with different side chains that you could build into a protein. They look just like our 20 amino acids, they just have a different side chain. Why aren't they ever put into a protein? There's no amino acyl tRNA synthase to hook them onto a tRNA. If you could synthetically hook them onto a tRNA, could you get them to go into a protein? Yes. You need to engineer the tRNA to pick a particular codon, though, right? So you'd have to engineer a tRNA to pick a particular pair, probably, of codons, and then synthetically attach that amino acid to that tRNA. And they've been doing that. So that basically you could come up and make a new protein using these engineered tRNAs that would have new properties never seen before in nature, because those amino acids aren't in proteins in nature. And what could they do? Maybe they could eat garbage or oil spills better, or get rid of nitrogens in the atmosphere, or sulfates in the atmosphere, or who knows what they could do. Maybe they could synthesize some kind of different chemical for us, an antibiotic difference or something. Who knows? That's pretty cool stuff. So I mentioned that it's universal, but there's a few differences, yeah? So in yeast, for example, and in mitochondria, for us, isoleucine is a start codon, or can be used as a start codon, as a methionine. So AUA actually encodes methionine in our mitochondria, but not in our nuclei, and in yeast mitochondria. Okay, this is in the mitochondria. UGA is normally a stop, but in mitochondria, it actually is a tryptophan. So you actually have 2 tryptophan codons in mitochondria, and you have 2 start codons in mitochondria. In our mitochondria, those 2 nucleotides for arginine, or codons for arginine, are actually stops. We also find that that's true in ciliates, which are little protozoans, and in yeast. So there's a few differences, okay? Don't need to know those differences unless you're working in mitochondria. You would need to know those differences, or if you're working in ciliated protozoans, you'd want to know those differences. So why does the 3rd nucleotide not matter? We talked this already. We said it's a wobble, okay? So I want to show you what some of the modifications can be on that particular nucleotide that can control the wobble and dictate specificity. So we've already talked about how G can base pair with U. So G can base pair with C, or G can base pair with U. C in the codon, in the anticodon, can only base pair with G in the codon. A can base pair with U. But there are instances of modified A's, which are really inosine, base pairing with C and A. Okay. These are some other modifications, right? If uracil can interact with G, these modifications actually prevent uracil from interacting with G, and only really allow it to interact with A. Okay, anything in a parentheses here was found in one example in nature, one example in one gene, not universally, but in one gene. So it could just be a sequencing error. They could have just made a mistake. But so this makes it kind of specific. How do those wobbles actually happen? Normally, you get 3 base pairs, 3 hydrogen bonds, right? Cytosine and guanine, 2 for adenine and uracil. When you have G base paired with U, you get 2 hydrogen bonds, but notice it's not the normal, like G wants to use all 3 of these positions right here, but it only gets to use 2 of them. It's not lined up properly, so it has to be wobbling. If you have a thiouracil, so thiouracil has a sulfur here, it can't make a GU base pair. It can only make an AU base pair. So that gives you specificity. That's this modification right here, right? So that modification only allows GC or AU. And then here's ionosine with C, with U, and it can go crazy and it can base pair with A. That's really weird to do that. So that just shows you what happens when we say wobble is really the nucleotides are in a different position. They really are wobbling, okay? And we looked at this before. Again, this shows you the tRNA anticodon sequence that can base pair to both codons. Okay. All right, so that ends up translation. So on Thursday, we'll talk about something completely different, as they say on Monty Python.